In today's episode, we'll be going through enzyme exam questions. The way I'm going to do this is that I'm going to show you from start to finish how I'm thinking, approaching, and answering the questions. And remember that the mark scheme is in the description box below. So let's begin. This particular question, I have a lot to say. This is something you really need to understand. It's a very common question actually, and not many students get it correct. You must firstly identify what's going on in the question, of course, but to actually understand what validity means. Now, validity is a concept in which you want the results to be valid. Valid meaning correct, reasonable, right? And for that to happen, there's a variety of things that need to be a part of the method, such as control variables, right? That's important because you need a way to standardize the method. If I do an experiment and you do an experiment, we both must have the same control variables for it to essentially turn out the same way, hopefully. Another thing would be you just need to minimize experimental error. And what I mean by that is about improving the accuracy or even the reproducibility and repeatability of the experiment. They are very important things you must mention and understand when it comes to validity. There are other things like control groups. If the experiment never said a control group or never mentioned one, say it. That's an improvement, right? Because you need something to compare it with the thing you're changing, or else how do you know whether it's changing or not? So there's all those different things you must think about. Now, if we read what's going on here, we can see that it tells you about the volume and the concentration of tripepsin solution, which is great actually. That's like a control variable. Next sentence tells you about the different temperatures used. So 10, 20, 30, 40, and 50. Now, to you or to someone else, it might be like, oh, that's good, They did a range of temperatures, but we're scientists here. We want specifics. The gap of 10 degrees Celsius is quite a lot. So an improvement would be take more intermediate values, right? Or temperature intervals must be closer together because it gives more of an accurate estimate of the optimum temperature. Because how do you know if the optimum temperature is happening at 24 or 26 degrees Celsius? You don't know unless you measure those temperature values. Another thing one could say is that there is no mention of a control group. And why is that important? Well, it's because to see if gelatin actually breaks down without the tripepsin. We don't know whether it does or doesn't. That's why we need to test that. There are other things like other mention of control variables. Yes, they said about the volume and the concentration tripepsin, but there's other things they could have mentioned, right? So, for example, the volume of the pH buffer. It only said, carry out each trial in the same pH buffer. How do I know, or how do you know how much you're meant to put in the pH buffer, right? You don't know. So that's why that needs to be specified. Or even the area of the gelatin. You need to talk about the volume, the thickness, the concentration of the gelatin. All of that matters. What if you use a different volume of gelatin for 10 degrees Celsius but use another for 20 degrees Celsius? Is that a method method standardized? No, it's not. So there's all these different things you must mention. Another thing is about, you can literally write this, and because it comes up in the mark scheme quite a lot, about ensuring that the method is standardized. I know I've been saying that word here and there, but it's true. And how can we enable that, right? How can we make sure this method is standardized? Well, measure the time for set volume of gelatin to be broken. What if you left one, when you're doing the experiment the first time, and you let the gelatin essentially get broken down for 5 minutes, but the next one you let it happen for about 15 minutes? Are the results going to be the same? No. This is the sort of thinking you need to have. is the method going to be the same if you do it differently and I do it differently? Or is the method going to be the same if we both do it the same way? Of course, it's going to be the latter. And hence why there's all these different points that one can mention, right? Sometimes this question comes up as not a 2-point sort of thing, because here it says, 2 improvements and then 2 explanations, but sometimes it can come up as 4 improvements and 4 explanations. Either way, the more the merrier, more points. It's very easy to identify these common pitfalls in the method. Why and how? Well, it's because we will continuously train ourselves to identify good methods and good experiments, and we'll see how they line up with, or don't line up, with bad experiments. And that all comes with practice. At first, I struggled with these questions so much until I realised it's the same things I'm going to be looking for each time the question appears. Next question says, suggest appropriate units to use to represent the rate of enzyme activity in this investigation. So we're going to go back to the previous page and look at the units that are used here. Now, we can see that there's volume and there's the percentage, essentially meaning concentration. But because we're talking about the rate of enzyme activity, rate meaning over a set period of time, right? So we know we need time as one of the units, but what's the other thing, right? So because we're measuring like the gelatin area meaning squared, so it'll be centimeters squared per minute, or you could say per second. Okay, next one says, evaluate the student's statement. So it's an evaluate, so I'm thinking yes, no. I'm not going to say about points of validity, only because it's a 2-mark and is too much. Let's read the statement. I think the mean is a more accurate measure than the median or mode for these results. Okay, so we have our focus, the median, the mode, and the mean. So that's the only thing we're going to look at on the table. Immediately, what just stands out is the fact that for isofit tripepsin, right, there's 2 modes. You don't need to be a mathematician to know that's just a bit, it's not very accurate, is it? It's either one or the other, but apparently here is both. They both show up the same amount of times. So that's a point that I could say about how the mean is more accurate, only because there's 2 modes that exist, and that in itself is the point. Another thing one can mention is if you do A-level maths, then you'll understand it a bit more than other people, but if you don't, no worries. It's the fact that with means, outliers are included when calculating the mean, or anomalies, or whatever you want to call it. And the fact that median or mode is less affected by outliers. So that's why the mean wouldn't be more accurate, actually it would be less accurate. Okay, over to the next question. It says, suggest the most appropriate statistical test for the student to use and explain why this test is appropriate. I don't even need to read this question or like anything else to know that the answer is going to be t-test. I know, that sounds like a bit of a stretch. How can you just say that? It's just that whenever the question is written like this, the answer's always been t-test for my revision. Of course, I wouldn't do this in the exam. I'd read the information, but because I've done so many questions, I just know that's going to be the t-test. Also, another hint that made me think that was because we were talking about mean in the question above, and so mean, I'm thinking t-test. If I didn't know that, I would read the information and I'd read the second sentence where it said they performed a statistical test on the mean of the 5 replicates for human tripepsin and the 5 replicates for the isofit tripepsin. The focus is means, and from all the statistical tests you have learnt in class, t-test is the one that you use to compare means, right? But there's one thing I really want you guys to understand. Please do not write and specify what sort of t-test it is. Don't say whether it is unpaired or paired. Reason being is that if you said the wrong one, then you're not going to get the mark. But if you just said, oh, t-test, that will guarantee you the mark. But if you said unpaired when it was paired t-test, sorry, you lost it. That's happened to me. That's why I'm like very passionate about it. Just make your life simple. I know you know it. Just don't risk it, please. Okay, and that's the end of the video. Remember, the two most important exam techniques are read the question first and state all the obvious points. Now over to you. Try some questions and let me know how it goes.